starting to record.
So one of the test yourself questions was how do centrioles look like flagella or cilia? And the only thing I could find in the book was that they have like molecular appendages, which help anchor them to microtubules.
Yeah, that would be correct.
Yeah.
Because the centriole is more of the base, is more of the initiation of the cilia and the flagella. The centrioles themselves are not causing mobility in the cell. So what you're saying, that sounds pretty accurate. I want to try this other webcam. I want to just try something real quick so that it doesn't look like. So then we've got the nucleus. And we talked about this yesterday. You know, oftentimes we talk about this being the brain of the cell. I mean, I guess that's the easiest descriptive. It doesn't function as our brain. Obviously, our cells don't have self-consciousness. They don't have a nervous system. It's not interpreting any kind of major data. What the nucleus is doing, though, is it contains all of the how-to. We've talked about this a million times. We have our DNA, which is just the code for how does this cell function and how does it react and interact with other cells and other structures within the body. Not all cells have a nucleus. You don't have to have one. Somebody remind me of the cell we talked about yesterday that does not have a nucleus.
Eukaryote?
Yeah, well, a prokaryote would not have a nucleus because pro before nucleus. Tell me a cell in your body that does not have a nucleus. I'll give you a hint. It's a transporting cell.
Red blood cells?
Nice. All right, use your med term. Tell me what the medical term for a red blood cell is.
Erythrocyte.
Very good. It's an erythrocyte. Very nice. So in this case, with the erythrocyte, a nucleus would really be counteractive. The whole goal, the whole role in your body of an erythrocyte is to transport stuff. It really is just like a little 18-wheeler tanker truck. Picks up a shipment of oxygen, drives it to the cell. At the cell, it picks up a shipment of carbon dioxide, drives it to the lungs, picks up a shipment of oxygen, and it just rinse and repeat. If my tanker truck was already half full with a nucleus, then I'm decreasing the carrying capacity. And in that case, the erythrocyte doesn't really need a nucleus because it has one singular job. It doesn't need to assess and it doesn't need to interact. One of the disadvantages, though, is that it does only have one purpose and it can never deviate from that. So it doesn't really have the ability to make decisions, so to speak. But the other thing about not having a nucleus means that the way that we produce red blood cells is different. Red blood cells do not produce or reproduce themselves by mitosis. They can't. They don't have a nucleus. Our body has to manufacture red blood cells and then pump them out into circulation. So it's kind of a disadvantage because that takes a lot longer than the act of mitosis. So that's where you could have conditions of like anemia, where your body is either underproducing red blood cells or destroying them faster than you can produce them. Here's our 4 parts of our nuclear anatomy. Here's our envelope, the nucleoplasm, which is just the jelly, our chromatin and our nucleoli. We talked about all this yesterday. Here's our nuclear envelope. Very similar, although not in structure, but in ideal, to our cell membrane. It's just simply contained. This outer layer of the nucleus, as we said, is continuous and continues on to our rough ER. The nucleoplasm is this jelly, very similar to the cytoplasm or cytosol, the jelly-like structure in the cell. Here we have our nucleoplasm, which is just a jelly in the nucleus. And this is where our DNA and RNA are, our DNA is contained, RNA is created. And what we have is this stuff called chromatin. And chromatin, again, I think we talked about this yesterday. Chromatin is little fibers that are made up of your DNA. And sometimes these little fibers will even come together or make exact copy replicas of each other. And that's known as a chromosome. And this is going to be important. You remember the typical kind of rounded X shape of a chromosome. And that's what we pull apart to make 2 exact copies. A nucleosome is just one single strand. Because remember, DNA is a double strand. I can't do a double helix. This is my interpretive dance of a double helix. You are welcome. So a single strand of DNA is a nucleosome. Yeah, this is new information, Hannah, this DNA, RNA, and chromatin. I know that at the end of class, I was just kind of mentioning the rounded X shape of a chromosome, but we did not specifically get to this slide. So during cell division, our chromatin, which is the fibers of DNA, they crunch down and condense, and they get what we call supercoiled, and then they develop that kind of typical X structure. Where's my pen? Where's my mouse? I'm losing my life here. Pen. So it kind of looks like.
When did they get supercoiled?
That would be during cell division. So during mitosis. So here's what's happening. Here's my cell, and I have all of these single strands of chromatin. And so what ends up happening is these single strands come, and so I'm going to obviously simplify this. Oh God, Nathan. Sorry guys, I just noticed, because I love this setup because I can see everybody right here. I have everybody on my screen. I have my PowerPoint down here, and I have a chat over here. And so if it looks like I'm negotiating between my 3 screens, and I just noticed what Nathan put on the chat. And you guys so you're not allowed to say anything about anything I say, because that was disgusting, Nathan. That was grosser than anything I've ever said. It's still funny though. Anyway, so with this chromatin, remember, we want to make an exact copy. So I have this chromatin, and what happens is it becomes super coiled, because I'm gonna need to compress it and then replicate it. Oh, Ace, if you really think that that is going to bother me, no. I got home, I'll tell you, let me finish this up and I'll tell you about the end of my day yesterday, where I came home literally covered in feces. It was terrible. So I'll tell you that. All right, so we have this super coil. And then what happens? So we've got a super coil organized now. Here I've got an exact copy super coil. And this is what makes that chromosome typical X shape. So that's what I mean by during cell division. We just need to simply compress. The other way to look at it is if you've ever recorded a Zoom meeting, my PowerPoint is another perfect example. What the program does is it compresses and uses less space for all my pictures in the PowerPoint. That's what's happening here with my chromosomes getting ready to divide. The nucleoli, this is where we are creating or transitioning to our ribosomal RNA. And this is also where ribosome subunits are made. And that's all we need to know about that. And that is chapter 3 in the bag. All right, so do you guys want to hear about my super bleepy day? All right, so this was, now that we've talked about all the parts of the cell, now we're going to talk about how does it work? How does all of this wonderful cellular stuff work? And so what we're going to specifically be referring to is fluid and proteins and things on the outside. How do they move to the inside and vice versa? So we have some terminology here, and this is just definitions.
Sorry.
No, go ahead.
This is chapter 4, correct?
It is chapter 4, cell physiology. So we have some definitions here. Intracellular fluid. Well, again, this is just med term, you guys. Should be super easy med term for you now. Intra, meaning inside. Cellular, the cell. Intracellular fluid is fluid found inside the cell. Extra means outside. So extracellular fluid means fluid outside the cell. And then interstitial, so again, inter means within. Stitial means tissue. So interstitial is fluid within a tissue. So this is how your medical terminology is going to be of use. Oh, Charles, I just caught a glimpse of you, my darling, and that is the cutest thing I've ever seen. That little pup just asleep on your shoulder. Very cute. All right, so those are just some definitions. Intracellular fluid, which is fluid that is found on the inside of the cell. Extracellular fluid, which is fluid found on the outside of the cell. And interstitial fluid is just fluid found within a tissue. Here's just some review of some terminology that we've covered previously. We got cation. Somebody tell me the charge of a cation.
Positive.
It's positive. And then, of course, our anion is negative. We have electrolytes, which are molecules that carry a charge. And these are all going to be super important when we're dealing with sick or injured animals. Electrolytes, some that carry a charge, some examples of molecules that function as electrolytes would be like potassium and sodium, calcium and phosphorus. All of these have very specific, very important jobs within the body. And so it's really important that we are able to monitor and replenish these electrolytes if we need to. So how do we get stuff in and out? And what is this stuff that we're trying to move? Now, this is different than the proteins that we've manufactured in the last chapter. What we're referring to here is the movement of things like fatty acids, amino acids that we can make proteins out of, different hormones. How do we get gases across the cell membrane? So this is a little bit different than the transport vesicles we just talked about. This is the movement of other structures. How do we get in the stuff that's going to keep this cell lovely and happy and healthy? And how do we get rid of the waste? So if you think about this excretion and absorption, remember we compare the cell to you. Okay, you are all now the equivalent of a cell. Well, what do we have to take in? We have to take in oxygen. We have to take in nutrition. You have to eat that Chipotle burrito. And then what do we have to do? We have to get rid of waste. So we breathe out gas waste. We poop and pee out solid waste. Same thing with your cell. Your cell is breathing, absorbing, and excreting gases, and it's taking in solid nutrition and poop. It's pooping. That's, I mean, if you ever want to just, we are all just walking sacks of cell poo. That's it. That's what a human is. We're walking sacks of cell poo. So it's kind of, yeah, we are all poop, Savannah. We are all poop. Everything poops, including your cells. Now, the process to get stuff in, like gas and nutrition, yeah, call that, yeah, Vicky, say you're just a walking sack of cell poo, which is the truth. So, see, this, you didn't think that your education here at the Vet Tech Institute would give you a whole host of insults to give to people, and they would have no idea that you're even insulting them. When I was in college learning medical terminology, we used to use medical terminology as insults. Call each other schmegma and anyway. Yeah, smooth brain. Smooth brain.
Sorry.
That's
That's all
funny.
it was.
Now all of you guys are just
Thank you.
shouting out insults. I love it. All right, so when we're taking in or removing gases, taking in and removing solids, two different
sorry.
processes that we can do this. The one process is passive. Passive means we do not need energy. We do not need the breaking of a phosphorus bond, ATP to ADP. We don't need that. Passive means no energy required. Active means we do need energy to do it. We do need to break that phosphorus bond in the ATP. So it kind of depends on the movement and what is it we're moving. What are we moving? That will determine, do I need energy to do it or do I not need energy to do it? So I've got a picture here, and I'm going to probably need to explain this picture because, you know, sometimes my pictures, you need an explanation in order to understand where Doug was on it.
Is that Red Rover?
This is Red Rover. I'm so excited that somebody recognized it. All right. So let's talk about the cell membrane. Remember the cell membrane, plasma membrane, this phospholipid bilayer. It's very, very complex, very, very complicated. And what the cell membrane's main job is to regulate what goes in and out of the cell. We already talked about this. The cell membrane is impermeable, meaning do not pass. You shall not pass. Impermeable to some stuff, and freely permeable, meaning free pass, everything come in. So I look at the cell membrane similar to, yes. Did someone have a question? Okay. Oh, mic is muted. Yeah, if you have a question, just unmute. No worries. Okay, so Red Rover reminds me very much of a cell membrane. So if we look at this little line of children, And if we continue that line in a circle, we now have, okay, this was not supposed to be there. We now have a cell membrane. So this cell membrane, if you, Red Rover, let whoever come over. And then this person goes running full tilt, and a couple of things are potentially going to happen. They're going to break through very easily, meaning that this cell membrane was permeable, very easily able to break through. So here's my, oops, no. Get my pen. So there's my cell membrane. So easily permeable. The second thing that could happen is that this does not allow it to penetrate, which would be impermeable. The third thing that could happen is this thing comes racing, running through, takes out a whole bunch of them, and the whole structure falls apart. That one is not optimal. But with Red Rover, that's usually what happened. Somebody always ended up crying. I'm just saying, it was a terrible game. It was a terrible, terrible game. But if you think about Red Rover, like if we're looking at active versus passive, do I have? No, there we go. So if I look right here, active versus passive, if this little guy right here breaks through, look, this guy's not ready. He is not ready. He's standing on one leg. He is laughing. He is not prepared. This little girl is not even holding hands. She is not, I know, just so weak. So what's going to happen? This little girl, though, look at she's got her guns out. She is ready. She's screaming at this guy. Oh my God, you need to get ready. So, no, he's not. No, he's not. So this kind of action, mental action, is really what I think of when I think of a cell membrane. How easily is a molecule able to penetrate the cell membrane, or is it unable to penetrate at all? So let's talk about some transport stuff. So we defined passive transport. Passive transport, remember, no ATP required. Yeah, so when I first, like years and years and years, a million years ago, started teaching anatomy and physiology, I made the very, very giant mistake when I was teaching the cell membrane and passive and active transport. I had the students go out into the hallway and we played Red Rover. And it got violent very quickly. And I am no longer allowed to do Red Rover. So, yeah.
That's what happened in our chapter as well. Like we played it for FFA one time and then they were just like, y'all aren't playing that anymore.
Yeah, I did it once and Mr. Hamilton just said, no, the angry parent phone calls are not worth it. You are not to be doing that again. And you can petition all you want, Britain, but guess what? It's my class and I can do what I want and I'm not putting anyone at risk. And we can't do it right now anyway because of stupid COVID. All right, passive transport. Let's talk about it. No energy required. We've got 4 ways of movement without using energy. These 4 ways, and we're going to go through each one of these separately. We have diffusion. We have something called facilitated diffusion, which is just simply we have a helper. Osmosis, which is the movement of fluid. And filtration, where I'm just basically like a coffee maker. I'm filtering through, and I'm using pressure. So these are the 4 passive transport, no energy required. So let's
What did you
talk
say
about
about osmosis?
osmosis is the movement of water.
Okay.
What about diffusion?
I'm literally going to start talking about that right now. So we're going to go through each of these. Oh my God, you guys, stop with the Red Rover. You're not playing Red Rover, not on campus. If you want to go off campus and play Red Rover, yeah, I know I started it, Charles. I know I started it, cheeky monkey. Yeah, in the parking lot after school. Oh my goodness. You guys make me giggle. All right. Talk about diffusion. So when we're talking about diffusion, not using energy, not needing energy, we have something called the concentration gradient. Now, this is a super, super complex idea that really is not complex. Concentration gradient simply means I've got an area of high concentration and an area of low concentration. And stuff moves from high to low. I'll give you a couple of mental images to give you an idea of the concentration gradient. So it's pre-COVID. I used to be a Texans fan. I think I'm a Cardinals fan now. I think I'm going to follow JJ and DeAndre. I think I'm going to start following Arizona. Anyway, jerk face, Easterby, not Cowgirls. No, can't follow the Cowgirls. But if you go to the game, huzzah. Inside the stadium is a super high, if you think of all the people as molecules, and there's super high concentration of molecules. Well, when the game lets out, all of the molecules they're not just staying where they are, they're automatically going from a concentrated to the parking lot, which is not concentrated. And so that's the whole idea of the gradient. You have high concentration, and the molecules are just automatically going to start moving into a low concentration. Here's another example of this high to low concentration. Bam. Yes, I just pulled this. Oh, I got to refill it. It's almost empty. Do you guys know what this is? This is beautiful. My mom got me this.
Bath and Body Works plugin.
It is a Bath and Body Works plugin. What do we call this? It starts with the letter D.
Diffuser.
Diffuser.
It's a diffuser. So that is exactly what we're talking about. If anybody else has a diffuser, it doesn't have to be Bath and Body Works. Oh, you were thinking liquid. No, not liquid. We're talking about the smell. It's a diffuser. So if anybody has something like this in your home, what is it when you put in a brand new smell? Have you ever done that? I usually try and stick with the same smell because then I am smell blind. But what happens to you when you put in a brand new smell? It smells good, but what else?
It's really strong.
It's very strong.
Oh my gosh, it is so strong. Especially right at the plug that you have this. That's a super high concentration of smell molecules. But then what happens? The smell molecules being super, super concentrated at the plug start automatically moving where? Throughout the room. So they've gone from an area
Less
of
concentrated.
high to low concentration. Low concentration meaning there are no smell molecules. And suddenly we now have a diluted smell. It's not nearly as strong. And so that is the whole idea and why they call this what? A diffuser. Because it is super high concentrated, automatically moving into a low concentrated area. So what are some things that can have an impact on this movement? Well, I mean, this is not rocket science. What's this? The size of the molecule. Small molecules are going to be much easier to move. Is the molecule able to pass through a fat layer, like the phospholipid bilayer? What kind of charge does it have? And temperature. There's a reason why you plug this in, because it, in fact, oh, that's kind of warm. It heats it up and causes it to diffuse faster. It also helps to convert the liquid into a gas, but that's another day. What we're talking about is the temperature causing it to diffuse faster. So those are the main factors that affect a molecule's ability to move passively via diffusion. And what are you guys LOLing about up here? No, you are not a rocket doctor. You guys, sometimes I wish when I'm in class on campus, I can read your, that's fine, Sierra. All right, so.
Can you go back for like 1 second, please?
Of course I can. So those are our determining factors. How easily and how effectively is this molecule going to move? All right, so when we're talking about the concentration gradient, remember it's just high to low. The difference between the two, the difference between the high and the low will determine how fast something moves. I love the analogy of the slide. Here I have the obviously the high concentration and the low concentration. And there's a really big difference between the high and the low, which means that you're going to slide very quickly down the slide. Here we have the high and the low, and we really don't have a whole lot of difference between the two, which equals the lamest slide ever. You're going to have to like push yourself down the slide. So that's what we're talking about when we refer to the concentration gradient. As molecules are moving, the speed with which they move will be dependent on the difference between the high and the low. And we're always, in this case with diffusion, we're always moving from high to low. And again, that makes sense to me mentally with the slide. Because if I am at the top of the slide and I push myself down, it takes no energy and no effort on my part to go. But if I'm at the bottom of the slide and I'm trying to climb up the slide, do you see how that is going to take energy on my part? That's a whole other thing. That is another process that we're going to talk about. Because there are some times that molecules will move opposite the gradient. But whenever we use the term concentration gradient, we are specifically referring high to low.
Miss Huff, and this is like the concentration is going to be high inside of the cell and it's going to move out, or it can also be from out to in?
Both. So let's take a look at the cell. let's say I have high concentration on the inside and low concentration on the outside, that's going to promote stuff to move out. If I have high concentration and low concentration, that would promote stuff to move in. And it's this idea that is so important for things like drugs, different medications. How do I deliver them to specific cells? Bam, this is one of the ways. So if I have a drug and it's targeting a specific cell, it's this gradient that promotes it to molecular move into the cell. There's a low concentration of drug inside the cell. There's a high concentration of drug outside, which means that the drug then moves into the cell. I know. So this is just another picture for you little visual learners. Here we plugged in our diffuser. All the little red dots are smell molecules. And then the smell molecules start to move throughout what we call this like a room until they are evenly dispersed. So smell, I'm actually going to stop recording.